I'm here to talk to you about cell structures and their functions. This lecture is not about the plasma membrane or transport across the plasma membrane. That is a separate lecture. So we're talking about primarily the structures inside a cell today. We're going to talk about prokaryotic cells briefly, just not in a lot of detail, but a little bit of detail. We're mostly focusing on eukaryotic cells in this lecture. First thing I want to note is that cells are not round. So you might have at some point had a biology class where cells were all drawn as circles on the board. Depending on what tissue type we're talking about, cells are all different shapes. So you'll take anatomy, hopefully, eventually, some of you. And if you do take anatomy, you will look at the different human tissue types. And you will learn that cells have all types of different shapes, depending on what that cell's job is, where it's found in the body. So the cell on the screen is not a round cell, right? It's not round at all. The nucleus is round, but it's also important to realize this is not a flat structure. It's three-dimensional, and it's not just this big gelatinous blob. There's a lot going on in the middle here. Two major categories of cells that exist in living organisms: prokaryotic cells and eukaryotic cells. And at the very beginning of the semester, we talked about what types of organisms are prokaryotes and what type are eukaryotes. Remember that term prokaryote really means before nucleus, and eukaryote means true nucleus. So that is the main difference between those two cell types. But we will get into a little bit more detail today about the major differences between those two cell types. You can see that included in the prokaryotes are bacteria and archaea. Whereas if you look at the eukaryotes, it was the animals, the plants, and the fungi. At the end of the term, we will talk about protists and fungi in more detail. We will talk about plants a little bit, especially when we talk about photosynthesis. So today we're mostly focusing on animal cells, although we will point out some important structural differences between plant and animal cells. A really important concept to understand is that cell size is limited. So when organisms were first evolving, they were all single-celled. The first organisms on Earth were single-celled. Single-celled organisms can only get a certain size. They can't get huge. And the reason for that is this concept of surface area to volume ratio. If materials are having to move through the cell by simple diffusion, okay, which we'll learn about in more detail when we get to the transport across cell membrane lecture. But the idea of diffusion is that materials move based on concentration gradient. So this is going to be movement of molecules from an area of higher concentration to an area of lower concentration. Sorry, that's concentration. So to area of lower concentration. So from high to low, and this happens without any energy required. Okay, so if I have a cell and I have a lot of a molecule outside the cell and just a little bit of that molecule inside the cell, these molecules will start moving into the cell by simple diffusion until equilibrium is reached. That really means equal on both sides of the membrane. So equal inside the cell and outside the cell. So that movement of molecules with no energy required is called diffusion. A lot of simple organisms move materials through their body and from the outside to the inside just based on simple diffusion. So if you have a really big cell, so let's look at this picture here. This is a pretty fat cell. If you look at the surface area, consider the surface area, imagine the whole outside of the cell in 3 dimensions all the way around. And then this area is the volume. Okay, so that's the volume on the inside. The whole outside is the surface area. If you're moving materials from the outside, you need a big surface area, but not a lot of volume. The more volume you have, the harder it's going to be to get those molecules from the outside all the way to the center. So they're going to diffuse in, but if you need them to get all the way to the center of the cell, that's going to take a long time if that's a fat cell. Versus look at these tiny cells. If you have molecules out here that need to move in by simple diffusion, that's not going to take long. So this is a very important concept. As cells get bigger, the volume increases faster than the surface area. In other words, that surface area to volume ratio decreases. You have less surface area compared to the volume. Cells can only get so big because of that, because cells are having to move materials around by diffusion. It's not possible if that cell gets too big. So the only way organisms were able to get larger is by becoming multicellular. If you have a lot of little cells, that's a better plan than having one big cell. You also can then start specializing those individual cells to perform different categories of function, and tissues can start to develop, and organisms can become more complex. So as organisms move from being single-celled to multicellular, they were able to get bigger for this reason. So this is a very, very important concept in biology. If you look at this picture and look at surface area to volume ratio, you can see we have this one tiny cell. Okay, so imagine this is a single-celled organism such as a bacteria. That bacteria can't get this big. That wouldn't work. Look at the surface area to volume ratio, it doubles. It would be really tough for materials to diffuse all the way to the center of that organism. But look, if we have something that's really the same size as this big block, but it's made of a lot of little blocks, each of those little blocks has the same surface area to volume ratio as this original. So this is a good plan. It just shows you organisms could get bigger by having a lot of little cells. And that's what organisms did. When they became multicellular, those cells were small. This really summarizes a little bit of what I said a minute ago. As the size of the cell increases, the volume increases faster than the surface area. And if you have too much volume, that means you can't move materials to the middle of the cell. Okay, things won't move as efficiently by diffusion if that cell has too much volume relative to the surface area. So that means when you do the math, if the surface area is smaller relative to the volume, then that surface area to volume ratio decreases. If you have a large surface area and a low volume, you have an increase in diffusion rate. So this would be small cells. Okay, so small cells have a large surface area and a small volume. Great diffusion rate. But as the surface area gets smaller relative to the volume, you have a decrease in diffusion rate. So this would be large cells. This is not a good plan. You actually have a lab activity based on this concept. So this is a very important concept to understand. The cell structure and function lab activity number 1 requires you to cut potatoes into little chunks, and those are going to simulate cells, and you're going to put them in iodine, and you're going to look at the diffusion rate of the iodine. So cell surface area to volume ratio is a very important concept to understand, not just in lecture, but you'll be demonstrating that in lab. So if you have questions about that, be sure to ask. Looking at entire organisms that are multicellular, this is also an important concept. So this is a flatworm. And flatworms are pretty primitive in structure. They don't have a vascular system to move materials around. Materials move mostly by diffusion. So gas exchange from the outside environment to the inside of the organism occurs a lot based on simple diffusion. And so this worm cannot get fat. So this worm has a large surface area, as you can see in the picture, a relatively large surface area and small volume. And that equals good diffusion. Materials are going to be able to move around more easily through this organism. These organisms cannot get fat. They can't get a big volume. They have to be flat in order to be efficient. More advanced worms, such as this earthworm, this is actually a very advanced worm. So this worm is segmented. He has specialization of those segments and has actually a very complex vascular system. Because of that vascular system, this worm is able to be fatter. Okay, so this worm has a lower surface area and larger volume. The only way this worm can achieve that is because it has a complex vascular system to move materials around. It's not just relying on simple diffusion. So that's another strategy that organisms used as they became more complex and started specializing different regions of their body to have a vascular system to move waste products around, move gases, nutrients, hormones, whatever needs to move through the bloodstream. That vascular system is going to enable that organism to become bigger and become more complex. Again, surface area to volume ratio, very important when looking at cells. Looking at eukaryotic versus prokaryotic cells, we've talked a little bit about the basic differences before. Remember, the big difference is a nucleus, but there are some other differences we're going to see also as we start looking. You can see already just looking at this picture that eukaryotic cell is much more complex in structure compared to that prokaryotic cell. We're going to look at that in more detail. Okay, so this is a basic prokaryotic cell. In fact, this is a bacteria cell. And the first thing that you'll notice is that this area called the nucleoid region replaces what would be called the nucleus in a eukaryotic cell. So this is the region where the cell has its DNA, but it is not protected in a membrane. You're going to see when we look at the eukaryotic cell that the nucleus is defined by what's called the nuclear envelope. So prokaryotic cells, and I'm just going to say prokaryotes, no nuclear envelope. The DNA is just free in the cell. Okay, a lot of students get confused and think prokaryotes don't have DNA. They have to have DNA to be a living organism. Okay, that's how they code for their proteins, just as more complex organisms do. So they have DNA, it's just not protected in a membrane. It's just free in the cell. And that area, that region where the DNA is located is called the nucleoid region. Also, no membranous organelles. Organelle means tiny organs. Okay, so when we look at the more complex eukaryotic cells, you're going to see a lot of organelles made of membrane. So they're called membranous organelles. Prokaryotes don't have membranous organelles. In fact, the only real internal structure that they have is they do have ribosomes. So only true organelle are the ribosomes. And the ribosomes are where proteins are assembled. So ribosomes, we talked about a little bit when we talked about nucleic acids. Ribosomes are where proteins are assembled. So right here you can see ribosomes. And these are organelles that synthesize proteins. This is the assembly plant for proteins in eukaryotes and prokaryotes. So if I were to draw a simple diagram of a bacterial cell that's a little less messy than that one. This, by the way, is not the shape of all bacteria. Okay, this is a rod-shaped bacterial cell. They're called bacilli. We'll learn more about the 3 possible shapes of bacterial cells later in the semester. Not all bacteria have this shape, but they all have on the outside here this plasma membrane. Which you'll see in eukaryotic cells also. Okay, and then they have this circular piece of DNA that wraps around itself a lot of times, that is basically one circular piece of DNA. And this is what's called the chromosomal DNA. This codes for all necessary proteins in the bacterial cell. Sorry, I just spelled that wrong. Chromosomal. Sorry, I'm really messing this up now. Chromosomal DNA. It codes for all the required proteins in that bacterial cell. The reason we make that distinction is because a lot of bacteria also have some additional DNA, a little ring of DNA, and they can have more than one. And this is called the plasmid DNA. And we'll talk about this more later in the semester. I'm just going to point it out now. Now, as I mentioned, the only other true organelle in the cell are the ribosomes. And I'm just going to draw those as little circles. No membranous organelles in this bacterial cell. Oh boy, sorry about that. As we get close to the edge, it just doesn't do well. I'll draw them here. Okay, and then this is important too. On the outside of that bacterial cell is a cell wall. Now, we learned about the cell wall when we talked about carbohydrates. Remember, the cell wall in plants is made of cellulose, which is a polysaccharide. It's a structural polysaccharide. So it's a chain of glucose that makes up the cell wall of plants. The cell wall of this bacterial cell, though, is made of something different. Okay, so here's the cell wall outside the plasma membrane. And when we talk about bacteria in more detail, you'll learn what that cell wall was made of later in the semester. Okay, and sometimes they have a flagellum to move, or sometimes multiple flagella for locomotion. So there are these tail-like structures. You can also have these little structures on the outside called pili. This is how they stick or attach to surfaces. So for example, that's how bacteria attach inside your digestive tract, in your colon, in your intestines, the bacteria that are really, really important to your gut health and your overall health. We'll learn a lot about your microbiome later in the semester, and that's how they attach, is with those pili. That's how bacteria attach to surfaces in your house, is with these little sticky structures. Okay, but otherwise very, very simple in structure. Some actually have a capsule outside the cell wall that makes them even more resilient. So things like anthrax and botulism. The reason those are so resilient is they have this capsule on the outside of the cell wall that makes them even tougher to kill. So they're resistant to UV radiation, heat, cold. So prokaryotes, very simple in structure, no nuclear envelope. The DNA is just free in the cell. They're all single-celled. No multicellular prokaryotes. If something is single-celled, it means the entire organism is made of just one cell. No membranous organelles. The only true organelles or tiny organs in the cell are the ribosomes. And those have the same function that they do in eukaryotes. They're involved in assembly of proteins. Plasma membrane, cell wall. That's it. Eukaryotes obviously are a lot more complex. Okay, so this is an animal cell and this is a plant cell, and you can see a lot more going on in these cells. We're going to talk about the similarities and differences just very briefly Okay, we're going to talk a little bit about the similarities and differences between animal cells and plant cells. For the most part, plant cells have all the same structures as animal cells overall. So the main ones we're going to talk about in this class are shared. One of the big differences is that plants have this structure called the chloroplast. And the chloroplast is where photosynthesis occurs. You're going to learn about photosynthesis in more detail. That's how plants take light energy from the sun and make carbohydrates to use for cellular respiration, because plants don't eat. So very, very important. Most important chemical reaction on Earth is photosynthesis. No life on Earth would exist without photosynthesis, and we will talk about that. Also, you see this plant cell has something called a vacuole. And that vacuole here is involved in water regulation, and animal cells don't have that. Plants need water to carry out photosynthesis. In fact, water is the first reactant that enters photosynthesis. So that is involved in water regulation in the plant cell. Those are two structures that animal cells do not have. Otherwise, all the main structures are the same. Rough and smooth ER, Golgi, ribosomes, lysosomes, mitochondria. Plants and animals have those structures. Plants, of course, have a cell wall, and you can see that cell wall on the outside of the plasma membrane in this plant cell. And remember, that cell wall is made of cellulose, which is a carbohydrate. It's a chain of glucose. It's a structural polysaccharide. Animal cells don't have that. So if we look at the list here of what they share, you can see that they both have all of these structures that we're going to talk about today. So don't worry if you don't know these terms yet. We're going to go through all of these today. Animal cells. No cell wall. We just talked about that. For the most part, no vacuole. Okay, no chloroplasts. And some animal cells can have a flagellum to move, but plant cells don't have that. Okay, let's start looking in more detail at the individual structures in the animal cell. Sorry, I dropped something. Hold on one second. Okay, sorry, I'm back. Okay, so this is the nucleus, and the nucleus of the cell is defined by the nuclear envelope that surrounds the nucleus. So the nucleus really is more of a, I like to think of it as a region of the cell, but it's a structure that's defined by the nuclear envelope. So if you look at a typical cell, just going to draw one here. In the center would be the nucleus. It doesn't have to be exactly centered though. And it's really the nuclear envelope that defines that nucleus. Okay, and then inside that nuclear envelope is the DNA. So that nuclear envelope is protecting the DNA from the rest of the cell, from all that chemistry happening out in the cell. This region outside the nucleus, between the nuclear envelope and the plasma membrane, is called the cytoplasm. And again, the cytoplasm is really more of a region of the cell. That's an L, sorry, it won't get bigger than that. So this whole region is the cytoplasm between the nuclear envelope and the plasma membrane. So we have the nucleus defined by the nuclear envelope, and then we have the cytoplasm. So if we go back to this big diagram, this whole region here, this would be the nucleus. Okay, and this whole region here is the cytoplasm. So there's a lot going on in the cytoplasm. That's where all of these other structures exist. So the nucleus is only just that small region, not exactly centered, but we usually say it's in the center of the cell. And again, it's defined by the nuclear envelope. And that's where the DNA is protected from the rest of the cell. Materials can move in and out of the nucleus through what are called nuclear pores. And these nuclear pores are openings in the nucleus, and you can see them here. All of these little nuclear pores, and here's one blown up. I love this electron micrograph over here showing the nuclear pores. Okay, so nuclear pores how materials enter and leave the nucleus. When we start talking about transcription and translation, you're going to see that messenger RNA is made in the nucleus. Okay, we make that copy of one gene in the nucleus, and then that messenger RNA leaves the nucleus through a nuclear pore and comes out to the cytoplasm, where a ribosome assembles around it to make the protein. So nuclear pores are how materials move in and out of the nucleus. You'll see that attached here is this membranous structure, and it's the first membranous structure we're going to talk about, called the endoplasmic reticulum. And you can see that it's contiguous really with a nuclear pore. So there's a lot of communication between the outside of the nucleus and the inside of the nucleus through these nuclear pores. But it's really the nuclear envelope that is defining that nucleus. And again, the DNA is all inside here, inside the nucleus, being protected from the rest of the cell. Ribosomes, again, we've talked about a little bit when we talked about proteins and nucleic acids, and we just talked about them a little bit when we talked about the bacterial cell. Now here they are. We have two types of ribosomes in a typical eukaryotic cell. They're what are called free ribosomes and what are called bound ribosomes. Okay, so the free ribosomes are just free in the cell, and you can see all of these free ribosomes out here. They're not attached to anything. The bound ribosomes are the ones that are connected to this membranous structure called the endoplasmic reticulum. It's abbreviated ER. Endoplasmic reticulum. It's a membranous structure that can take one of two forms that we're going to talk about in a minute. But the difference in these two ribosomes is significant. They're both involved in protein assembly. So this is where the messenger RNA recipe is read in the ribosome and is translated into the amino acid sequence of the protein. Okay, so this is where all of the amino acids are strung together to eventually make that polypeptide chain that folds into the final protein. The difference is that free ribosomes that are just free in the cell, this is where we assemble proteins that remain in the cell. Okay, so for example, key enzymes that are needed for that cell to carry out its chemistry. All of its chemical reactions require enzymes, and we make those enzymes on free ribosomes that are just loose in the cell, free in the cell, not attached to ER. We also have what is called bound ribosomes, or what are called bound ribosomes. And these bound ribosomes are attached to what's called the rough ER, the rough endoplasmic reticulum. Those bound ribosomes are attached to the rough endoplasmic reticulum. And this is actually a pretty good setup, and I'm going to show you why. So if this is a typical cell, And here's the nucleus. And then attached to this nucleus, we have some endoplasmic reticulum, membranous sacs, and on the endoplasmic reticulum, we have some ribosomes. Okay, remember there's some free ribosomes in the cell. That's where we're going to make proteins that stay in the cell. But then attached to this membranous ER are some other ribosomes. If we blow up that ER, I'm going to blow it up bigger. Sorry. Blow up that ER right here. Here are the little ribosomes on the outside. What can happen is the cell can start making a protein. Make it green. It can start assembling a protein, and that protein can start feeding into the inside of the ER. So here's the protein. It's getting assembled here. I'm just going to make a little blob. And now a piece of this ER can pinch off, carrying that protein product. Okay, so here it's going to pinch off. And now it's going to be this separate little sac, and it's going to be carrying that protein product here in the middle. There's our protein in the middle. This is a very handy setup if we need to export that protein. So those bound ribosomes, those are involved in assembly of proteins for export. Let me erase this part. So attached to rough ER, and they're involved in assembly of proteins for export. Again, this is a really good setup. The protein, as it's being translated, feeds into the lumen here. A piece of that membrane pinches off, carrying that protein product, and now that can move eventually to the plasma membrane and leave the cell. And we're going to see how that happens. So if a protein needs to be exported, it's going to be made on the ER, and it's going to be packaged in that piece of membrane. A membranous sac carrying a product in the cell that pinches off from the ER or the Golgi or another membranous structure. So here's that membranous sac, and it's carrying that protein product. So here's the protein inside there. This is called a transport vesicle. A vesicle is just a membranous sac, a sac made of membrane. So vesicle is a membranous sac, a sac made of membrane. And if it's a transport vesicle, it means it's a vesicle carrying a cell product. That transport vesicle just pinched off from the rough ER, carrying that protein product. So that's a very handy setup. We're going to see what happens later to that product. Okay, this is a ribosome blown up, by the way. It's not just this little circle. You can see there's a large subunit and a small subunit, and you're going to see that the messenger RNA is going to feed through this ribosome and get translated into a protein. And you'll see how that happens later in the semester. Okay, so free ribosomes, bound ribosomes. The bound ribosomes are on the rough ER. So that is the function of the rough ER. It's involved in protein assembly for proteins that get exported from the cell. So for example, we've talked about insulin. Okay, insulin is a protein hormone. Insulin is made in the pancreas. Doesn't do us any good if it stays in the pancreas. It needs to get out of those pancreatic cells and into the bloodstream to allow us to regulate our blood glucose levels. The way that happens is it's made on rough ER in those pancreatic cells, and then is packaged in a transport vesicle, pinches off from the ER, and that's going to move through the cell, and eventually that product is going to leave the cell. And we'll see how that happens. There's another type of ER called smooth ER. So you can see here in this picture, the rough ER has ribosomes. The smooth ER does not have ribosomes. Smooth ER, in fact, has completely different functions. I'm just going to quickly tell you about a couple of functions of the smooth ER. So the smooth ER does not have ribosomes. It looks smooth under the microscope versus the rough ER looks rough because it's covered in those ribosomes. So functions of the smooth ER. Okay, the smooth ER is involved in detoxification. So drugs, alcohol, other toxins that come into the body, the smooth ER in certain cells of the body detoxify. They attach functional groups to those molecules that allow them to be more easily flushed out of the body. They make them more water soluble, so we get rid of them more easily. If you take a certain medication or you drink alcohol over a long period of time, you develop more smooth ER and you get better and better at detoxification, and you develop what's called drug tolerance. And that is because of the smooth ER. So, think back to if you are 21 and you drink alcohol, the first couple of times you drink alcohol, you feel it, right? But over time you build tolerance, and that's because of your smooth ER. So your cells are going to make more smooth ER and get better at detoxifying. And then if you stop drinking, your body is going to recycle what it's not using. So a lot of that smooth ER in the cell will get recycled. The parts will get broken down, used to build other structures. And over time, you'll kind of go back to that same set point again. But detoxification is an important function of the smooth ER. So what organ do you think in the body would have cells that have a lot of smooth ER? Where in the body do you do most of your detoxification? It's in your on your screen. Yep, it's about to. There we go. That's so strange. So liver cells are involved in detoxification. They would have a lot of smooth ER. By the way, you can tell a lot about a cell's job by looking at the amount of organelles of a certain type that it has. So cells don't have what they don't need. So a cell packed full of rough ER would be a cell that's really involved in making a lot of proteins for export. So cells of the pancreas that make insulin or glucagon, those are protein hormones made for export. Those would be packed full of rough ER. Cells of the liver that are involved in detoxification would be packed full of smooth ER. So that's one function of the smooth ER. I'm not going to give you all the functions today. Lipid synthesis. Okay, assembling certain types of lipids. Okay, so this is where phospholipids would be assembled. This is where the lipid-based, cholesterol-based hormones are assembled. And remember that included cortisol, our stress hormone. It included estrogen, testosterone, progesterone, so the sex hormones. Okay, so cells of the ovaries that make estrogen would be packed full of smooth ER. Whatever that cell's job is, it's going to have a lot of that organelle to perform that job. So cells of the ovaries that make estrogen would be packed full of smooth ER. Cells of the testes that make testosterone would be packed full of smooth ER. Okay, I'm just going to give you one more function of the smooth ER, even though there are more than this. Third function, sorry, this slide is getting really messy. Third function, calcium ion storage and release. And this is really involved in muscle contraction. So your muscle cells have a lot of smooth ER. In fact, the smooth ER of the muscle cells actually has a special name. It's called the sarcoplasmic reticulum. And this is the smooth ER of your muscle cells. If you go on to take anatomy and or physiology, you'll learn about the sarcoplasmic reticulum. So that's a third function of the smooth ER. Again, totally different functions than the rough ER. Different enzymes embedded in that membrane. But just as a product of the rough ER can pinch off in a transport vesicle, the same thing happens in the smooth ER. Okay, so let's say this is our smooth ER, and let's say this is in the ovaries, and a lot of estrogen hormone is made, and now it needs to get out of the cell. A piece of that will pinch off into a transport vesicle, carrying that hormone product, just as we saw with the rough ER. Okay, the Golgi. This is another membranous structure in the cell. This is a stack of membranes that are really involved in processing products that were made in the ER. So you can see this transport vesicle right here carrying a product, and it carries it to the Golgi, and now that product enters the Golgi, and it's going to move through these stacks of the Golgi. I really messed that up where you can't see it very well. It's going to move through the stacks of the Golgi, and it's eventually going to come out the other side. So there's kind of a receiving side of the Golgi and a shipping side of the Golgi. But what happens is those products get modified. So the Golgi is involved in modification of products. And eventually labeling those products for export. This is sometimes known as kind of the UPS center of the cell, where those products come out the other side in a new transport vesicle, and now they have some sort of tag telling them where to go. There are some other important product functions of the Golgi, but just know that they're involved in product modification and labeling for export. We won't talk in detail about the other functions of the Golgi. One more membranous organelle that I'm going to tell you about in a minute, but I'm actually going to skip that slide and go to this picture. All of these membranes in the cell work together through what's called the endo, meaning inside the cell, endomembrane system. You can see the nuclear envelope, the rough ER, the smooth ER, those transport vesicles, the Golgi, all work together, and eventually the plasma membrane is involved too. And what's important to realize, because we're going to see this theme in particular when we look at the plasma membrane, it's important to realize that membrane fuses with other membrane. Remember that membrane is mostly phospholipids, and that doesn't just include the membrane on the outside of our cells, the plasma membrane. It also includes all of these membranous organelles. So whenever we say membrane, we're talking about mostly phospholipids with some key proteins involved too, but a lot of phospholipids. And those membranes can all fuse with each other. So that's how that transport vesicle can pinch off from the rough or smooth ER carrying a product. It can fuse with the membrane of the Golgi. And then a new transport vesicle can pinch off the other side, as you see here. And then you see it moves to the surface of the cell where it fuses with the plasma membrane. And here we see that that's going to open up and release that product out of the cell. I'll go ahead and give you this term now. We're going to talk about it in more detail in the video about the plasma membrane. And that term is exocytosis. And this is materials leaving the cell, okay, through fusion of a transport vesicle with the plasma membrane. So that transport vesicle moves to the plasma membrane and those materials leave the cell. So that could be a protein that was assembled on the rough ER. It could be a hormone, such as cortisol, estrogen, testosterone, that was made in the smooth ER. And that transport vesicle is going to carry that to the plasma membrane, and then it's going to leave the cell through what's called exocytosis. You're going to learn that we can also bring materials into the cell by wrapping plasma membrane around, bringing it in through what's called endocytosis. So when we learn about transport across the cell membrane, you're going to see endocytosis is the opposite. That's wrapping something in membrane and bringing it in. So the endomembrane system is incredibly important in the cell. The one structure I didn't tell you about are the lysosomes, and lysosomes have a lot of really important functions. But one thing that they do is they recycle old cell parts. So all those structures in your cell have a lifespan. We want to reuse those parts to build other structures. So those all get digested. So really the lysosomes are a sac of hydrolytic enzymes. Do you remember what hydrolytic means? We talked about hydrolytic enzymes when we talked about pH and the digestive system. And remember in that intro to organic chemistry lecture, we learned about a specific chemical reaction that breaks down big molecules into smaller molecules. And remember that was called hydrolysis. So hydrolytic enzymes carry out hydrolysis. So what lysosomes do is, you can see here, the lysosome is in purple, and it's going to fuse with this old mitochondrion that's being carried by transport vesicle. Look, membrane fuses with membrane. So this transport vesicle is made of membrane, so is the lysosome. They fuse together, and then those hydrolytic enzymes are going to digest that other cell structure and then spit out the parts. Lysosomes are actually involved in cellular eating as well. So single-celled organisms such as protists, they use lysosomes to digest their food. So you can see here, food comes in, it gets wrapped in membrane. So this would be endocytosis. It gets brought into the cell, wrapped in membrane. Membrane fuses with membrane. So this membrane is going to fuse with the lysosome. And the lysosome is actually full of hydrolytic enzymes that are going to digest those food particles. This is also how certain of our white blood cells work. So we have some white blood cells called phagocytic white blood cells. You'll learn what phagocytosis means. It's a term that's a very specific form of endocytosis, but those phagocytic white blood cells use lysosomes to digest pathogens. So the lysosomes are going to be an important part of your white blood cells being defensive. In other words, breaking down foreign particles, dust, dog hair, things we breathe in. So we'll have phagocytic cells in our lungs that break down those foreign materials. They can actually kill bacteria and other pathogens that get in, parasitic worms, okay, through either emitting different chemicals, but there's another way that certain white blood cells do where they actually devour. It's called cellular eating. They actually bring in that foreign cell so they can devour a bacterial cell and use their lysosomes to break that down. So if you get a splinter in your finger, bacteria get into your skin, these phagocytic white blood cells are going to come devour that bacteria and use their lysosomes. We also use lysosomes to do something that's called apoptosis. It's spelled apoptosis. And this is when entire cells digest themselves. So there are certain times when an entire cell would need to digest itself. If there's a DNA mutation, if that cell has been invaded by a virus, we're going to learn about different ways that that happens later in the semester, but that is one of the roles of the lysosomes, is digest that entire cell. When you're developing, you actually have webbing between your fingers, and lysosomes in those cells of your skin are signaled to digest those skin cells that are between your digits. So at some point, that webbing is digested. Some people have a genetic mutation that allows some of that webbing to stay. And you can look up those pictures on the internet if you're curious. Okay, so lysosomes, it's a sac of hydrolytic enzymes. It can digest old cell organelles. It can be used for cellular eating, including in our white blood cells, and it can be used to digest entire cells. If we need to destroy an entire cell, it's called apoptosis. Another term for apoptosis is cell suicide, where all the lysosomes open up and digest that entire cell. Okay, lysosomes, very important. I know this is a long lecture. We're getting close. Mitochondria. Okay, these are not part of the endomembrane system. In fact, mitochondria are pretty unique. This is where most of our ATP is generated during cellular respiration. So mitochondria are where most of our cellular respiration takes place. This is where ATP is produced. So mitochondria are involved in ATP production through cell respiration. You're going to learn a lot about that. You're going to learn about the detailed structures of the mitochondria. There's an inner membrane, an outer membrane, a matrix, an intermembrane space. You don't need to know those structures now. The reason they're just shown on this diagram is realize when we learn about cell respiration, different stages of cell respiration are going to take part in different regions of the mitochondria. So there is some complexity of structure there that you will need to know in the future. Mitochondria are really, really unique. They act almost as independent organisms. So let's talk a little bit about what those unique characteristics are that make them like independent organisms. But I also want to point out that mitochondria have their own ribosomes. They have their own mitochondrial DNA. And if you look at this mitochondrion, not only does it look like a bacterial cell, but it actually functions a lot like a bacterial cell. And it's pretty well understood in biology today that mitochondria used to be independent organisms. And they were taken in by other bacterial cells and became symbiotic. It's called the Endosymbiosis Theory. And it applies to both mitochondria and chloroplasts. So you can see an ancestral eukaryotic cell that really wasn't able to very efficiently produce energy. You're going to see that some of our ATP is made in the cytoplasm, but most of it is made in the mitochondria. And at some point, these bacteria were ingested by the cell, and now there's this symbiotic relationship between these structures that used to be free living bacteria. So rather than the cell digesting them, it allowed these bacteria to live in the cell, and now they serve really important functions. So really the endosymbiosis theory includes mitochondria. Sorry. Every time I touch this with my hand, it does something crazy, and it's impossible to write on the screen without touching it with my hand. And I know there's a setting where you make it only work with the pencil, and I've got that turned on and it still does this. So mitochondria and chloroplasts are part of the endosymbiosis theory. They both function as independent organisms. They have their own DNA. They divide independently from the rest of the cell. Mitochondria even have their own ribosomes, so they make their own proteins. Let's talk a little bit about that. Okay, so if you look at that mitochondrial structure, we mentioned before that they have their own DNA. It's called mitochondrial DNA. And we're going to talk more about that in a second. They have their own ribosomes. And so they can make their own proteins. Unique features. They divide independently, independent of the rest of the cell. They make ATP, so essentially they can make their own energy. Okay, obviously they have DNA and they can make their own proteins. That really meets the definition of a living organism. If you think back to the very beginning of the semester when we talk about what makes a living organism, mitochondria act just like living organisms. And in fact, that mitochondrial DNA looks very much like bacterial DNA and encodes for a lot of the same traits. So we're very sure this endosymbiosis theory, to reach the level of theory in science, remember, that's a big deal. That means it's been supported by considerable evidence. Lots of data has been collected by many, many, many people over many, many, many years to reach the level of theory. So it's pretty cool. Also important to realize that you get your mitochondrial DNA only from mom. Okay, your chromosomal DNA, half is from mom and half is from dad, but you get your mitochondrial DNA only from mom. So when egg and sperm unite, here's the egg. And in the nucleus of the egg, you have 23 chromosomes. And out here are the mitochondria. I'm just going to make little squiggles in the middle. So here are your mitochondria in the egg. Here comes the sperm. Okay, it also has 23 chromosomes in the nucleus. But guess where the mitochondria are on the sperm? They're mostly all on the tail, and they don't become part of that first cell. So when those two fuse, they form that very first cell of 46. Okay, 23 from the egg that made you and 23 from the sperm. And all the mitochondria of that very first cell came from the egg. And this divides and divides and divides to make grown-up you. And every single one of your cells has mitochondria only from the egg. So all from mom. So all mitochondria are from mom, which means all of your mitochondrial DNA is from mom. If you've ever watched a crime show, you know you can leave mitochondrial DNA just as you can chromosomal DNA. Obviously, every single cell has millions of mitochondria. So mitochondrial DNA is easier to find sometimes than chromosomal DNA. The only way someone on this planet has your same chromosomal DNA is if you have an identical twin. Otherwise, everyone's chromosomal DNA is slightly different. But we have a lot of similarity in our mitochondrial DNA, because if you think back to the very first Homo sapiens, that same mitochondrial DNA has been passed down generation to generation to generation. You know, they have what's called the mitochondrial Eve. Whoever the first human is that gave us our mitochondria, we all have really similar mitochondrial DNA. Now, obviously, there have been some mutations over time that have occurred, but for the most part, it's very similar. And that's true for all animals. And so you can trace relationships among animals based on similarity of their mitochondrial DNA. So evolutionary biologists use mitochondrial DNA to trace relationships among organisms. So that's pretty cool too. Mitochondrial DNA is really, really interesting. And you can see it's circular DNA, just like bacteria have. Okay, chloroplasts you're going to learn a lot about when we talk about photosynthesis. So this is only in photosynthetic organisms. And it's not just plants, okay? Photosynthesis occurs in certain photosynthetic bacteria that have chloroplasts. It includes the photosynthetic protists that have chloroplasts, and of course it includes the plants. So plants aren't the only cells that have chloroplasts. We're going to mostly talk about the plants, okay? But there are photosynthetic bacteria and photosynthetic protists that came before the photosynthetic multicellular plants. Plants are multicellular. Okay, protists and bacteria are all single-celled. The difference is that protists are eukaryotes and bacteria are prokaryotes. But these chloroplasts are where photosynthesis occurs in all of these different organisms. Sorry, this says multicellular, but again, toward the edge. I don't even know why I haven't learned yet to not write toward the edge, because you just can't read it. Sorry. Peroxisomes are an organelle in certain cells. I only have this on here because we have an enzyme lab later in the semester that involves peroxisomes. Sorry, I'm just going to quickly plug this in because I'm getting the signal that my iPad is getting low. Peroxisomes carry out an important reaction that we're going to study in the enzyme lab. And what peroxisomes do is they take hydrogen peroxide, which is a product of a lot of chemical reactions in the cell, H2O2, and they convert it to water and oxygen. Water and oxygen, okay, not toxic, obviously. You have a lot of water and oxygen in your body and it's not toxic. Okay, this is hydrogen peroxide and it is toxic. So peroxisomes are involved in making that conversion from this toxic hydrogen peroxide. And if the peroxisomes are working, they have an enzyme called catalase that breaks down that hydrogen peroxide into water and oxygen. And you'll see the oxygen makes bubbles. And that's why when you put hydrogen peroxide on your skin, it causes bubbles, because the catalase in your cells is breaking down that hydrogen peroxide into water and oxygen. And it actually is destroying the membrane of any bacteria that are there. So that's why we use it. But you'll see those bubbles as the oxygen is released. So again, the only reason I have these peroxisomes on here is because catalase enzyme and hydrogen peroxide, we're going to study that in the enzyme lab. We're going to actually use yeast. And yeast have catalase enzyme and can do this same thing. We're going to look at hydrogen peroxide and how it's processed by yeast. The cytoskeleton is important to understand because we're going to look at different functions of the cytoskeleton, especially when we study cell division. There are several elements of the cytoskeleton. So if you look at this slide, you can see there are what are called microtubules. There are microfilaments and intermediate filaments. Really, the only one you need to specifically know the name of are the microtubules right here. And that's because when we talk about cell division, the microtubules are actually going to pull the two sister chromatids apart. Or if we're talking about meiosis, they're going to pull the homologous chromosomes apart and eventually the sister chromatids apart. So the microtubules, which are part of the cytoskeleton, are going to be involved in cell division, which is one of the functions of the cytoskeleton. But as far as microfilaments and intermediate filaments, you don't need to necessarily know those terms. But what I do want you to know is I want you to know kind of a list of functions of the cytoskeleton. So the cytoskeleton is really filling the whole interior of the cell that's not filled by those other organelles. And so looking at functions of the cytoskeleton, I want you to realize there's not really a whole lot of free space in the cell. Things don't like free float like gelatinous blobs through a cell. They move on this transportation network called the cytoskeleton. So let's look at some different functions of the cytoskeleton. And I'll probably have to erase a couple times because I don't have much room here. Cell structure and shape are one of the functions of the cytoskeleton. The cytoskeleton is what gives a cell its specific shape and really holds that structure and shape. It also anchors the organelles in place. So nothing is really free floating in the cell. It's the transportation network. So for those transport vesicles, they don't free float from the ER to the Golgi. They move along this transportation network called the cytoskeleton. And then when those transport vesicles need to move to the plasma membrane to get materials out of the cell or move materials into the cell, those materials all move, those transport vesicles move along elements of the cytoskeleton. Okay, they're involved in, so this would be movement of materials inside the cell. They're also involved in movement of entire cells. Okay, so flagella are extensions of the cytoskeleton. And as we said before, involved in cell division. And you're going to see how that happens. That's the microtubules in particular. There are other roles of the cytoskeleton, but that's a pretty good list of what you should know. And again, you don't need to know, well, the microfilaments do this and the intermediate filaments do this. Just know that these are some functions of the cytoskeleton. Okay, that's finally it for the cell.